everyone. So I'll start my session today. I'll just go through assignment 1 that was given. I think it's already been done. I'll just give some insights and hints about assignment 1. So the first question is, when a vertebrate animal, horse, dog, etc., leaps up or gallops, what do you mean by leaps up or gallops? So they suddenly can move very fast, and let's say they hear a very loud noise and they react and starts running. So which of the energies directly acting on the motion that need to be considered for a balance of energies? Which is the main energies? Ignore those that are too small to make difference to the size scale. So here they're asking that which is the energy that gets converted somehow so that they can gallop. So first of all, gravitational, you can just rule out gravitational and muscle energy. Thermal energy is not the correct answer. You can just by intuition it says it has to be mechanical energy that's getting converted. Next is photosynthesis involves which process? It's pretty simple. It's interchange of energy. So what happens there is they change they get thermal energy and they convert it to chemical energy that is stored in the leaves via chlorophyll. So what is the scale of these cells? Typical bacteria of E. coli under choose the right order of magnitude. You can just see as far as I remember, the E. coli has the structure of that around micrometer order, 10 to the power 6 micrometer, so that is 10 to the minus 6 meter order. So HIV is also, you can just look up, that is 10 to the minus 7 meter approximately. What is the scale of this green fluorescent protein? Choose order of magnitude. So green fluorescent protein GFP, so you know that it is 730 base pair. So this is another way. So this is a length scale unit used for proteins / DNA, etc. So what is the base pair? As you know, for every DNA structure there is this AT-GC bonding, hydrogen bond is formed. AT is adenine, thymine, cytosine and guanine. So they make one base pair. So if it is 730 base pair long and one base pair is approximately 3.4 angstrom. So you can calculate the whatever the total length of this GFP, green fluorescent protein. One important trivia I'll tell you: GFP is a nice marker for cells to detect, like it's a green fluorescent, and on a microscope you can see that there is this green green dots that represent cells or nucleus, whatever you want to detect. Next, what is the osmotic pressure gradient acting on model E. coli? So osmotic pressure gradient model E. coli suspended in pure water. Given the concentration of osmolytics, the model bacterial all the, so what is given? Given is concentration is given, volume of the E. coli cell is given, and the temperature. So for osmotic pressure there is this empirical formula. So this C is the concentration R is the ideal gas constant, T is the absolute temperature, T is the absolute temperature. So ideal gas constant, and this I is called Van 't Hoff factor, Van 't Hoff factor for it. So it is defined as number of formula units initially dissolved in the solution by over number of particles in solution after dissociation. So usually for non-electrolytes, this I is equal to 1, sorry, I is equal to 1, and ideal gas constant value you all know. So if you put all of this together, you can calculate. As far as I remember it, I calculated it approximately 2 atm around the values came. So which of the following drives the diffusion of molecule in solution in the absence of concentration gradient? So this is a very nice question. So what is the diffusion of molecules? So if you see, if you have, let's say, a solvent, solution like this, and you have full of solvent like this. Now you add some molecule, let's say you added salt or sugar, or let's say in your room you spray some perfume. So what happens is, eventually if you wait long enough, this high if you diffuse it, molecules diffuse from a higher concentration to a lower concentration and region, and eventually the whole thing, like let's say initially the molecules are like this, highly concentrated. Let me change it a little, like this. Let's say it was around here, and eventually if via diffusion the whole thing homogenizes. What do I mean by homogenizing? Homogenizing means what do I mean by homogenizing? Homogenizing means that if the concentration of these molecules will be even throughout the region, so it will become more like this. So if you calculate the concentration around this surface, around this solvent, this concentration will have a gradient. So there is a higher concentration here, there is a higher concentration here, and there is lower concentration here. So what happens is, in diffusion, concentration travels from higher concentration to lower concentration, so high to low. This is what is called molecules are going from concentration gradient. But after it homogenizes, so the question is, which of the following drives diffusion of molecules in the absence of of concentration gradients. So if there is no concentration gradient, what happens in this scenario? They are asking what happens in this scenario. So here there is no concentration gradient, so there will be no more homogenizing left. So this concentration, so from this travel from higher concentration to lower concentration happen by chemical potential, and only thing that can move, that can make the molecules move, is this, if it is in a temperature bath. So let's say if you have a temperature heat bath here around all of this, so it has a finite temperature. So the temperature here of this system, if not equal to 0, so they always have a kinetic energy and they can move. So that can drive drive the diffusion. If you have a absolute 0 temperature, there is no diffusion happening. So thermal energy is the only answer. The number of cells with time of asexually dividing cells growing by binary fission. One important point to note here is binary fission can be represented by which of the following expressions? Symbols TD is the doubling time and T time is NT. So what is binary fission? Binary fission is nothing but from one cell dividing into two cells. So eventually this also leads to two more, and also this leads to two, this is binary fission. So happens is this is 1, 2, 4, 8. This is how they grow in number. As you see, so at every, and TD is the doubling time. So what is the doubling time? So the time required for the cell to get doubled than the population it started with. So if at T equal to TD, NT should be 2. So NT is, if at T equal 0 NT is 1, then let me write it properly. If at T equals 0 NT is 1, then at T equals TD NT should be 2. So what is NT? NT is the population of the cells at time t. Now if you look at these solutions, this C solution, it has NT equals 2 to the power T by TD. So at T equals 0, NT becomes 2 to the power 0, which is 1. At T equals TD, NT equals TD is 2 to the power 1, which is 2. So this is the correct answer. Next question, and minimally how many membrane bilayers will an imaginary line drawn from a fibroblast to the longest axis of the cells and passing through its genomic center? So for a fibroblast, this is a schematic of this fibroblast. So if you draw a line like this from its longest axis, so this might seem to be symmetric, this exactly schematic, but it is not. It has some kind of a bias towards one axis. So whichever direction you go, you see there is, if you don't hit the Golgi body, you must hit the nucleus. So this nucleus has one bilayer here, and when you are passing out, there will be another bilayer. So the right answer will be 2. So what are the lengths of the E. coli and human embryo? Still again, it's you can look it up. Probably it's either of these two. So the answer will be B. So this concludes the week 1 assignment. And so this I will delete. Question 5. Yeah, most of these questions from week 1 was most of the questions from week 1 was based on some information that we must not know. There is not much to to teach here. So this will be the answer, and this is week assignment 2 for the week 2. Which molecular properties of water? So most all of this I am not going to give you answers of this. So most of these questions have answers in the main lecture videos. If you go through it, at least as I can remember, this question, and this question, these 3 questions are exactly calculated. It is there in the main videos. I can give you some insights here. Sorry. So if you go to the videos here,
next to water in terms of the chemistry.
So here he is discussing how the hydrogen bond is formed in water and what are the angles and all, and eventually if you go boiling point, freezing point, those are the first questions. What is of the water directly responsible for unusual properties of water relating to surface tension and boiling point? So the first question is, which of the molecular properties of water are directly responsible for unusual properties of water relating to surface tension and boiling point as compared to chemically similar compounds? So surface tension and boiling point related answers you will get here. Here, so boiling point, unusual boiling point, what are the, so this is the surface tension. You see this here, the insect can walk on the top of water because of surface tension, and this related answer he is discussing here. This is the boiling point, freezing point, everything is unusual because of the hydrogen bond and some other reasons that was discussed in this lecture. Next question comes is what aspects of molecular water get is cryopreservation, freezing. So for those who don't know cryopreservation, he also has discussed cryopreservation. So cryopreservation is a process where the cells are frozen and kept from dying. Now frozen and preserved. Now after when you need the cell to regrow or culture, you can bring back from the cryoprotectants. So there are specific process how to do it and what are the causes related to this, and he has discussed everything here. How the cryopreservation around this part, you will know how to do the cryopreservation, what are the important points to be noted for the cryopreserving. So, and then comes this part, Bernal, Fowler and Pauling model for ice. So this part is also very important to understand ice and water, so which here he has discussed here. So this is the model of tetrahedron model which is given by Bernal, Fowler, and the structure and entropy of a crystal. This is this Pauling paper, and you can also go through the papers of this Bernal and Fowler paper. These are available online. So what are the exact structures? It's like this, and so in detail he has discussed. If you go through this, you will be able to answer all of these questions. So question number 3. So question number 4 says explain how the water molecule in the 3D geometry proposed in Bernard Furling model achieves a combination of six possible conformations. So this is a very interesting question where these 6 possible conformations are given here and how it is. So to answer it, you will have to go
Say that
this
tetrahedron
lecture.
that you see on the left.
So there are like 4 possible states and 2 of the molecules can rearrange themselves. From there this NC2. So this whole thing you have to answer, you have to write to get the full marks here. So the effect of hydrophobicity. What is hydrophobicity? Hydrophobic means those who hate water, like they don't want to bond with water. Now let's say O2, oxygen molecule, you have to put in water. Now what is the free energy, and if it is energetically favorable it will bond, if it's not it would not. So this question also has been calculated by him here, hydrophobicity and entropy. So this is a red one vertex of this tetrahedral is occupied by a molecule, and he exactly goes on to calculate from this portion exactly goes on this to calculate the entropy. And entropy, the concepts you need to know is entropy gives free energy these two, and you have to calculate which one is the entropically favorable and which one is not. So here you can see this G equals TH minus TS. So S enthalpy and yes. So S, if you go through your thermodynamics properly, so you will know this S is Kb log omega. Okay, so it says log W. So W is the number of ways the molecules can be arranged. Number of ways molecules can be arranged. So you have to calculate W of both cases without the oxygen and with the oxygen, and you get this difference minus Kb log omega. So from this difference you can calculate delta S, and from delta S you can calculate delta G and show in the units of Kb. This whole problem has been done in the lecture series. You have to go to the lecture series. That concludes the discussion about assignments. So if you want to look at the previous lectures, so from here you can go from this weekly log sheet. There is this YouTube link where if you go in this link you can actually access the previous year
2024
last
3
week's
p.m. to 5 p.m.
discussion here. where we have discussed about viscoelasticity and how shear thinning and shear thickening molecules are, and then we have discussed Stokes law of viscosity and how to calculate the viscosity of water and other fluids via a ball drop experiment. And here we have discussed oscillatory shear flow. To get the link, you can go here in this the PDF. So this is the classwork PDF. This has been done, and this week's also will be uploaded here.